Welcome back, everyone, from coffee break. So my name is Liz Carlisle, and in the spirit of our international meeting here, Leonard is tag teaming to me here for the second half. I am from Stanford University in California, and it's really a great pleasure to get to moderate this afternoon and to be part of this meeting this week. So the first folks we're going to hear from in this second segment are two leading plant breeders working in perennial agriculture. So we're going to hear first from Lee DeHaan, who is lead scientist in the Kernza Domestication program at the Land Institute, leading the effort to develop intermediate wheatgrass into a perennial grain crop. And then we'll hear from Fengyi Hu, who is Dean of Agriculture at Yunnan University in China, where he leads a growing program in perennial rice breeding. So I'll turn it over to Lee and then Fengyi.
All right, so thanks to you for the introduction and invitation to speak to you today. Fengyi and I are going to kind of contrast the two big approaches that we often talk about in developing perennial grain crops, wide hybridization and domestication, although I'm going to throw a little bit of a wrench in that, and I don't think those two paths are quite as clear as we might at first think. But by and large, what I have led has been a domestication program, taking a wild crop, wild plant, and turning it into hopefully a grain crop. Kernza is the name under which we sell the seed of intermediate wheatgrass. And just a little brief history on where this crop has come from. Began at about 1990 with breeding at the USDA and at the Big Flats Plant Materials Center, and that was in cooperation with the Rodale Research Institute from Pennsylvania. They did 2 rounds of selection until 2000. I started working at the Land Institute in 2001, and they had just dropped that program at that point with some changes in administration. And so we were able to pick up that seed about 2001 to 2002. I was starting to play around with it, but it wasn't really my main job. I was actually working on developing perennial wheat by wide hybridization. This was my side project until 2010. At that point, I had mentioned to Wes, hey, I think this can actually go somewhere. How about we hire someone else to take over the perennial wheat? We did that. And so I've been on this full-time since 2010. But in 2010, there was only, as far as I know, me basically working on intermediate wheatgrass, and everything else that's happened has been in the 8 to 9 years since then, and it's been really exciting to see just how many partners and how many people all over the world. I can't put up my list of collaborators because I'd fill the whole slide. So everyone, it's been exciting to be on this journey and just to see how many people are picking up this work. I'm going to try to talk about just the breeding side. There's a whole lot more involved in domestication about how do we grow the plant. Many of you are working on that. We've heard a lot about that this week. But just from the breeding side, I'm going to talk about, here's my outline to show you where we're going. First of all, just looking at domestication versus wide hybridization. And so on the left, we have how I see domestication. We often talk about it as this sort of funnel of weeding out the stuff we don't want. So we start with a big, diverse gene pool in a species, and we got to kind of narrow it down to something that's going to be the useful set of genes to make our domestic crop. We do that with cycles of breeding over and over and over. We pick the best ones, mate them together, and plant them out again. With wide hybridization, we're trying to hopefully find a bit of a shortcut by using a domestic species, a domestic annual crop, and crossing it with a wild species. And you see I have 2 different size arrows here, and this is because some of the projects we have are kind of like half and half. They're really not just shooting a little perenniality in. They're really kind of mixing it all together, and we end up with something that's kind of halfway in between, in a sense. Or, in the case of perennial rice, we actually pretty much have rice with a little bit of genetics from a perennial related species. And then we can end up with something that looks a whole lot like, functions a whole lot like in the diet and even in agriculture, like the annual crop. But we might have cases, as I showed with the wider arrow, where we end up kind of something totally new. We might have not one or the other. It's going to be something that we've never seen before. Maybe perennial wheat ends up that way. I don't know. So these are kind of 2 parallel directions in perennial grain crop domestication. So if we development. So with domestication, Intermediate wheatgrass was a forage grass in the United States. It's been used as a forage grass throughout Europe, in fact, Eurasia. And so it wasn't like we had to take something that was of no use to humans and make it domestic. It was already being used by humans. We're trying to add on a new use, which is, can we get human edible grain out of this crop alongside forage, which in some ways lowers the bar. So we're not trying to make wheat that just lives forever or lives a long time. We're trying to take a useful crop and make it even more useful. So the entry point, we hope, can be maybe a little easier. Right about 2008, 2009, we grew a field of about 30 acres near Salina, Kansas, and were able to harvest it with a standard combine and even get a whole truck full of seed using conventional equipment. So this kind of was a conventional crop in a sense. Farmers have to grow the seed to use it as a forage crop. It has to be grown and the seed harvested, so we actually know a fair bit about seed production. So what we're entering into then is a breeding process where we're going to do selection, evaluate plants, mate them together. So that all hinges on basically transfer of pollen, as Wes likes to say. So we're taking pollen from one plant and finding other plants that we like, and the white fuzzy things are the stigmas. The female parts of the grasses are often kind of ignored. Extremely important. So the pollen goes to there, and we get hopefully something new happening. We've been controlling that with a highly technical term of paper bags, the plant breeder's best tool. So paper bags and twisty ties, we like to say, that's about what you need. And you can control where the pollen goes and hopefully develop something truly new. So an example of something I did was just selecting for seed size over and over. So this is pretty much every year we're able to do one cycle, select the plants with the biggest seed, intermate them, just go on and on and on. And in 12 years, you can see we'd more than doubled seed size. People talk about how far do you have to go? Well, to equal wheat's seed size, on this trajectory, it would be 55 more years to get to the size of wheat. So we're going to talk a little bit later about some shortcuts, perhaps. I was missing hopefully I got the right PowerPoint here. Missing a slide that was going to transition to some of the traits that we're talking about. So a lot of things involved in domestication. The big one is often shattering. Already been mentioned here today. The plant holding onto its seed so that we can come by and harvest it and not just have that seed laying on the ground. So plants that we found like this one on the left, I just took my hand and ran it over this plant and kind of one soft brush and all that seed piled up on the left. I did the same thing to the plant here on the right, and you can see the seed is 100% of it still staying there. So we actually have the genes in this species. We started with actually at Rodale, USDA started with only 14 individuals, and those 14 plants apparently had enough, if we take the perspective of this morning, enough broken genes basically to already be able to come up with plants that don't shatter without having to break any more of them ourselves or have to search for them. We can just start making crosses between the least shattering plants, and in 5 generations or so, we come up with plants that don't shatter their seeds at all. It looks like there's a lot more than one gene involved in that in the species. We have a hexaploid, so it has a lot of different places for a gene to be, and then lots of versions of genes that are involved in this. Our very best plants, even after you thresh them, that rachis doesn't break apart. So there's lots of places to break in a head. So you can shatter with all different points. So it takes actually a lot of genes to stop shattering. It's not just one simple thing you can go in and fix. It's fairly complicated. Our best plants really hold together, and you get this nice nice seed. The next thing is naked seed. So to get that hull off, these ones on the left you see have a hull still on them. That, as was pointed out, that's what nature likes, protect the seed. We want that off so we're not trying to hull a truckload of hulls, fluffy hulls to town, or trying to eat those hulls. And so to get them off is quite a chore. We found genes that apparently control this. Plants that have more than 98% naked seed we found. It's been associated with the same gene that controlled free threshing and nakedness in wheat, the Q gene. Same thing seems to function here, but again, many more genes are needed than just that one. Takes quite a number. Seed size is something that we've focused on. It's important for domestication, easy to handle bigger seeds. In the greenhouse, we have seeds like this growing right out of the heads. So selection over time has actually released these plants from their constraint in their seed size. So in the greenhouse, I can grow 25 milligram seeds, almost as big as wheat. We take the same plant to the field, and suddenly we get 10 milligram seeds. So there's a lot more genes involved in getting a big seed than just sort of that release. That's step number one. Step number two is breeding over time, cycles and cycles, to change that plant to be able to fill that seed. So it's not just the simple, maybe there's one or two genes that control keeping the seed size small. We can find those easily enough. The next step, many generations of selection that will still take many, many years. And hopefully, all these new combinations come together to get us a plant that we can actually establish in the field. Things like seedling vigor being important to domestication. I did use the wrong. Oh well. Okay. So we're on now point the shortcut genomic selection. So how might we accelerate breeding? That's not right. Oh yeah, use genes from crops. Sorry, I wish I had the right PowerPoint. Okay. One idea is to be able to use that shortcut to shoot in a few genes, maybe from annual crops. So we're still kind of taking this domestication route, but what if we can cross to some plant like wheat and not try to like merge them all together completely, which gets all kinds of complexity involved in that, but just to, like we do with our annual crops today, we bring in a few resistance genes from the wild perennial. Maybe we can bring in a few yield genes or reduced height genes or free threshing genes from annual wheat. And so we've done this over the last several years, and it involves a process such as this. So it might be difficult to make that cross. We can take that young embryo and make sure that it lives. Something that is rare in nature, now we can make it common and start to bring in those genes. And so I have a pretty big program on this right now, and we're hoping that it might cause a breakthrough and give us the ability to take a shortcut. The next thing is genomic selection. So maybe we can accelerate breeding by using genomic selection. So in this approach, we would not have to grow plants out in the field. If we knew, based on little bits of DNA we could look at, if we could predict the best plants without having to grow them for many years in the field, we could actually, it's sort of like genetic testing for humans. Maybe you could go and test how tall you're going to be if you have a young child. You might be able to predict that from the genes that we can see. Same way here, if we can get a good enough model, we can make that prediction of the best plant. So we kind of go through, this is the current pathway that we're taking. The catch here is you actually need to build that model. So you do need to take some plants and grow them up in the field. These are the plants in the greenhouse, seedlings that we can take DNA from and make selections. But off the side, we have another program of evaluating plants in the field where maybe they can spend 3 or 4 years in the field. So this is great for perennials. We can evaluate plants for many years and develop this model based on performance as perennials, but then cycle the system of breeding very, very quickly. So maybe we can go 3 or 4 times faster using this approach. We've been doing now a couple cycles of this. Jerry Crane has talked about it here and is intimately involved in helping me to do this. So here's an example of what we call the training population to train that model. And the plants on the top are 2 years old, and then the next ones are young plants. And normally I would have been just selecting on these young plants every year because I didn't want to spend the time to let them grow up. I wanted to select every single year. In this case, now we can actually have old plants, have young plants, and evaluate them at every stage and develop that very robust model of performance. So this genomic selection really seems to be something that's just made for perennials. It's being tried in every crop right now, but everyone suggests, perennials is where it's really going to pay off. Finally, or second to final, we'll talk about the how we're going to market crops that are on the way to domestication. I think it's a little bit different than if you have a wide hybridization program and you produce rice that no one can tell is any different than rice. You can just put it right into the rice market. If you're domesticating something new, we have to have a new approach. It's not going to perform just like wheat, perhaps. One of the easy ways into this is to keep your percentage in any product low. So I worked with the baker early on. This is back in 2008 or 9, and he baked a loaf of bread. He said, I'm the pickiest person out there. I can't find anything wrong with this loaf that has 20% Kernza in it. Tastes fantastic, functions great. You try to go up to 100%, he wouldn't say that anymore. So there's sort of a low bar of taking something new into our diet. If it can substitute at a fraction, and in fact actually make it, in some cases, taste better and have a special flavor that we can enjoy, we can increase the value of that marketed product. Next approach is similar, putting it into a highly developed processed product that we can tell a story on the can, as they did with the long root ale, coming out with Patagonia Provisions. This is another sort of entry point into the market that doesn't require something that's a direct substitution for an existing crop. And we've had numerous beers come out after this one. So something that can actually be done quite easily, even on a very small scale. This North Cold beer, they told me they wanted to have a beer come out. We got them some grain in December, and they had that beer produced within 3 months in the can on the shelves, from concept to product, just like that. It's kind of a new world in beer today, and so it's been a good fit for us to get the story out there. All right. And because this is the wrong presentation, how am I doing on time? Who's my timekeeper? Where are we at?
Ready for Fengyi.
All right, great. Fengyi, you're next. Who's going to talk about? But I didn't get to my conclusion, which I should have done. But so I'll let Fengyi go ahead, bring up your presentation. I'll try to get a conclusion made. So I think this domestication is unique in the fact that we can kind of ease into, perhaps, especially if we can use the crop we're domesticating as cattle feed. If it has an existing use, we can lower the bar so that we don't have to have yields that are exactly the same, or functions in food that are exactly the same as existing crop. That's the case in which I think domestication is going to work well. We're going to see it as a forage crop that we also harvest a bit of grain from. That's our entry point right now. And hopefully in the next 10, 20 years, we get our yields up to being the same as our current domestic crops. Fengyi.
Okay, it's time for me to introduce the other way for developing the perennial crops. As just Dr. Lee said, the domestication method is very important for humans. And 10,000 years ago, our ancestors used this wild to domesticate the most wild plants and wild animals to cultivate the crops, and also for many animals for food for us. Okay, it's my first time to Lund University. Also, it's my pleasure to have this opportunity to share our job. It's a long time, 22 years ago, and my group start for perennial rice development. Okay, it's my. At the beginning, in the Land Institute, as we know, set up in 1976. As this, I think, is should be forced for many these problems during this modern agriculture. I think this too many scientists talking about this. What happened in the modern agriculture. Okay, I just have an example for agriculture, like this. I think it's a different agri-system between Asia or South Asia and Africa, because this area is more small-sized farm for agriculture, for food production. Like this. This is in the Yunnan province in the south of Yunnan. And most is upland rice for this farmer food. And as we see, this land, and after harvest the last season, and all this land exposed in the sunlight whole winter. And after next year, when next year the rain is coming, so it's led to having the soil erosion. And someone and most people think about, if we have some material covered in the land after harvest of the upland rice, it should be reduced erosion, soil erosion. Okay. Also, other bigger problem for rice production is for hard work for this job, like this. And even we have cattle to help these people for rice production, but I think this farmer should be put hard work for his food. And the other one is terrace. Terrace rice field is very popular in the mountain all over the world. So like this is very short, about 1 or 2 meters. But this cattle and the people, the farmer, should be cultivated the field for rice food production. So this is hard work for rice production in Asia, or I think in Africa like this. Okay, what is annual rice? There usually we have 6 steps for annual rice production, seed purchase, and I think for most farmers should be do. And for second is seeding nursery, and transplanting, and soil preparation, and transplanting, and so on, too many. But this annual rice, even harvest 1 time or twice, and some regional, maybe we harvest 3 times. But in the overwinter, the plant should be die. So each year, the farmer should be and recycle these 6 steps. Then, based on these situations, so most people think about, can we use rhizome, similar wild weeds in the rice field, and use the rhizome trait to developing perennial upland rice to balance food security and environment benefits for this region. We can see too many scientists, also is me include. And other important, very important from IRI, as we know, International Rice Research Institute in Philippines, in 1989, initiated a project, Frontino project for perennial upland rice project. It's funded by BMZ in Germany. And I'm lucky. I have this opportunity to join this meeting. When I'm young, I spent my master degree at IRI. So this is my supervisor, Dr. Eric Sachs, now have a job in the UIUC. And this David, Lee DeHaan, Landward from Queensland University, Stan and Weiss, also from US. And I think in 2010 visited us. We see this is a sloping and this upland rice. So initially this think about developing the perennial rice for food security and environment benefits. Okay, this is how to developing this perennial rice. It's more important how we can get the trait rhizome. Totally the 22 wild species of Oryza. And if like Dr. Lee, we can use the different wild species of rice to use the domestication method to developing upland rice or perennial rice. But we can find just only the 4 species and pulse this rhizome. And for other species, this different genome with sativa. This is rice, cultivar rice. So only longistaminata and the similar genome with rice. Okay, this is longistaminata, Oryza longistaminata, originating from Africa. And we see this is a very strong rhizome. And this dig out from soil. If we wash the soil like this, it's very strong. And so we use this material as a parents hybrid with sativa. And it's my idea. So can we merge this rhizome to annual rice and develop wild rice? This think about very simply, but actually it's too hard work. Yeah. Okay. This is for 1997. We crossed this ID23, crossed this wild species. This is a very famous cultivar rice developing in Thailand and also widely used in South Asia, also in Yunnan province. So we get F1. And after this one, we wield this very conventional or traditional this breeding method, self-cross, back-cross, this mass, and molecular assisted system selection for breeding. This is, we get the first, this F1. It's my supervisor also in the years before I have a job in Yunnan Agriculture Academy, Yunnan Academy of Agriculture Science. Okay. Why is different method for developing perennial crops? Because for this F1 is very difficult to get it. And according to this reference up to now, so far it's just a few case for get this F1 because this wild hybrid species hybrid is very difficult. We should use this embryo rescue method get this F1. After crossing maybe 1 or 2 days, this young emperor should be die. Okay, based this F1, how we for this the perennial rice. First we see this picture. If this perennial rice successful, so for the production we just need 2 steps, and for this 4th we no need for rice farmer. Before I said it's a very hard work job for the perennial rice developing, and from 1997 up to now, I feel the scientific papers published in the different journal, but each papers we have this one question to be solved. This first one we found this F1, and this PNS and this 2 this both, and for this rhizome gene QT mapping and major gene mapping. And based this mapping, we use the master for quickly selection from larger population, even F2 or this different generation. We get more different this breeding lengths. And this science paper, I think most of all of us joined this paper, right? And this one is my PhD papers and how to find this gene, and we sharp up this field markers, and then this will de novo sequence this wild species for this phenomapping, and this both this papers is for PR23 released in the China also trials both the China and the Laos. Okay, this for example, this in the PNS we found this two genes. This is one of the major gene and the other one major gene. Also, the too many cotiers control this rhizom's length. How many rhizome? This how long is rhizome? How interlopes between the rhizome. So it means rhizome developing the traits controlled by 12 cotiers in the right genome. Okay. The more important is what is the priority of rice. We see this is PR23. After harvest, this next year, the young tiller is emerged from and grow this rhizome. This is more clear. Like this. This is very short, this rhizome here. This wild species of Longis terminata. So why spend so long, 20 years more? Because this during 20 years, we also short this rhizome for like this, and we dig out too many cotiers in a different breeding lengths. Because if different cotier and this rhizome is very longer, is for survival the ability is very useful, but for rice production is not so good. And for example, for crop management, we cannot control this population of individuals in the field. Okay, we can compare the other one. This is we see the resulting annual rice. Some region in the, especially in tropical region, this also is the tooling for this rice. After cutting, and can we get next second season's harvest like this? But we see this all this heading from this bud above this ground, this bud. So this is a difference between the perennial rice and annual rice. And for annual rice, all this harvest from above the ground. But for perennial rice, all this tiller from underground. Okay, this is so far is my group's bigger progress. I think is for all of these perennial crops, and we get this PR23. This is first released in Yunnan province, means released by government, so it can commercial to farmer or some seed companies or rice food production companies. I think this is a milestone for not only for perennial rice and also for perennial grain. And if someone who is interested in this cultivated, you can go to the website to see in details. But I'm sorry, maybe it's in Chinese. Okay, but I can translation. Okay, and this other one is PR107. Is a new perennial breeding length. I think it should be released next year, and is under trials this year, last year, and this year. I think almost learning the Lam Jimmy yesterday introduced in the Uganda testing is very suitable for Uganda for rice production in future. Okay, this always my group is job. Okay, I will show us quickly this for case in the Yunnan province for perennial 23 demonstration. This is from 2016 to now. Just go quickly. This is first season. It's first season we also similar annual rice and use seed to necessary to transplant into for this field prepared similar. That's first one. The second after harvest, first season harvest, and we can see this benchmark here is a banana tree, and I think most of the Land Institute scientists visited there before. Okay, this is 1 year, but it's second season, and this is over winter. I think it's in the December. I take picture in this field. Something like this is hopeless for the perennial rice. But in the next year, over winter, once spring and this temperature is increased slowly, so we can see this new tiller is emerged from the soil. Okay, this is second year's first season. We miss third season, and this is fourth. Yeah, this fourth season. So this after fourth season, and we can see something here. Weight is a very, very important problem for perennial rice. I think also for other perennial ground production. Okay, this is the first seasons. This is 2018. This fourth season. Okay, this is a harvest. This is the February. We take picture in the this field, and this is now the end of last month. We take picture. It's new. It's similar. This I think it's similar. First season. This look like this. It's successful for third year, but as tomorrow my postdoc Dr. Yi Jiao said, how many years will be go? I don't know. We don't know also. I think this year is no problem. What about 5th year? So we waiting for this time passed. Okay, and we can see if the annual rice successful for perennial rice, for this fourth steps we should be 0. But how? What is the benefits for us, both for food and the environment? We can see we have some investigation. You see, the seed necessary transplanter for this annual rice. This I think this is important. And for perennial rice is first season, but for second year. So this 0. Also for this lab import, this 0 is 0. So actually we can calculate is just half for perennial rice for second year. This also similar. We combined this input like this. This income for farmers. How this just half, nearly half. Okay. We can see here, it's served per hectare per seed. This means the China yen. It's also for labors inputs, and I think it's more important. Just team show us this last before this first day we visited the companies. This employee is very decreased quickly and up to now from 400. Okay, 400 more to 229, right? So also in the mostly in the countryside, the farmer, the labor is decreased very quickly. So food will be for rice production is an important question for us. Okay, if we compare this year, we can see this first season, the this first year, second year. We combine the first season is similar, and the second season is similar because the different season, this yield is different, but it's similar annual rise in this region. Okay, we both this yield income for this library served for farmers. Okay, finally, if this perennial is successful, so we can solve this different this questions for rice production. So we think this rice for better world is true. It should be true, and before I said is a transformation from annual to perennials for sustainable agriculture. However, there are more challenging for us for perennial rice like this. This is genetics we don't know very clear, and the dry tolerance, how to cool, and the grain quantity, the diversity of cultivars used to distribute all over the world, and the crop management, and also too many. But the last one is more important, the funding for us, right? So this opportunity also too many opportunities waiting for us. Okay, up to now we this perennial rice, perennial 23 also distribute all of the world. This in China, this in Southeast Asia, in Africa, in Uganda. Maybe this year we test in the Katotiwa, maybe Indonesia or Australia. Okay, perennial rice to go well for sustainable agriculture. This is for 20 years more. We get more fund from different this funding. FAO, Land Institute, and too many is now I moved to Yunnan University 3 years ago. Also going for forever for perennial rice. Thank you for all.